Today we have Dr. Hao Zhu from Tulane University, and he's going to talk about interpretable AI, data-driven and mechanical modeling for chemical toxicity and drug safety evaluation. Dr. Hao Zhu is a professor in the Division of Biomedical Informatics Genomics. And before he joined Tulane, he spent a number of years in the Rutgers and working on toxicity prediction, preclinical translational research quite extensively. He's very well established informatician in this field. Today, he's going to present this chemical toxicity and drug safety evaluation topic. Welcome to Dr. Zhu. I'm sorry. Yeah, go ahead.
Thank you for your introduction. I'm very happy to be here to share a project that has been ongoing in my lab for close to 10 years with you. And also by the end of my presentation, I also want to briefly introduce our Center of Transporters. So I'm a modeling person, so I like to use informatics tools, data analytics, to perform a different kind of modeling studies for chemical topics. So chemical toxicity is one of the major studies in my lab. Let me see. Yeah, so today I have three parts of my talk. Okay, so first introduce some background, and then I wanted to introduce an old project that has been finished in my lab like several years ago, but still very, I think it's pretty interesting. So in the end, I wanted to introduce interpretable modeling, which is the major research in the recent like 3 years. Okay, so first, let's talk about some background knowledge. So I always want to start my talk with this picture. Okay, so what is the nature of machine intelligence? A long time ago, people saw a bird flying on the sky. So they wanted to simulate this process. Okay, so you may see that this guy standing in front. So he saw that this long beak was the reason for bird flying. So he gonna jump from the cliff and he gonna die. So obviously this is a negative experiment, okay? But this guy standing behind him was his mentor. So he gonna go back to the cave and talk to other professors. And they gonna send other students or postdocs to keep doing a similar experiment, but not the same. And eventually they're gonna find out that the wings are the reason for bird fly. But during this procedure, people would not repeat the same mistake they made before. Okay, so the power of language was to communicate, but also to record what happened before and perform certain analytic to avoid same mistakes. But this is also the most powerful ability of computers. So this picture actually came from a paper published 15 years ago, not very new, but still pretty good to describe the current chemical toxicity evaluation in 21st century. Okay, so on the left side of this picture, you may see that traditionally people evaluate the chemical toxicity using human, so this is in clinical trial, or using lots of animals. This is preclinical studies. Okay, but in vivo chemical toxicity evaluations are pretty expensive and also low throughput. Okay, so we spend like millions of dollars just to evaluate one chemical. So this is unaffordable. So in the past decade, scientists majorly focusing on designing alternative models. So they use low-level animals. In the middle of the picture, you may see that they use fish, they use bee, they use worms. And the most popular method to do so is to use high throughput screening methods. Okay, so all these alternative models, they can be used to screen, to assess a chemical quite rapidly. But unfortunately, okay, so you see that on the bottom of this picture, the throughput of this all these alternative protocols increase, but however, the relevance to human, to animals, this is what we really care about, also decrease. Okay, So in the middle of this picture, this is the position of computational toxicology. So traditionally, we saw that the role of computational models is to directly predict the chemical toxicity to human, to animals, or we can prioritize, select the highly toxic chemicals for animal studies. But in the past decade, just due to the development of all these alternative models, we gathered huge amount of toxicity data. So in a so-called big data array, the new role of computational toxicology is to make good use of this big data coming from this all these toxicity testing assays and make even better models for human or for animals. So this is the strategy of QSAR. Okay, QSAR refers to quantitative structure-activity relationship. It was an old concept. It was designed like probably 40 years ago. Okay, so if you want to perform a QSAR study, first you need to have a database consists of quite a lot of chemicals. Okay, instead of link the chemical structures to the target activity or toxicity on the right, you need to calculate chemical descriptors. This is a quantitative value to represent the chemical structures. Then using machine learning algorithm to link the chemical descriptors to the target toxicity or activity. Okay, so the strategy is still quite applicable or useful for some simple chemical properties or activities, but unfortunately, for complicated chemical toxicity, it won't work. Okay, so what's the reason? It showed here is based on the theory of QSAR, a similar chemical will behave similarly in bioactivity or toxicity testing, right? So I intentionally select these 4 chemicals. So they consist of the same chemical scaffold. And if you just look at the above 2 chemicals, okay, so these 2 chemicals are really similar. So the only difference is on the left, upper left corner. Okay, so this chemical and this chemical has a slight difference. But if you perform animal testing for these 2 chemicals, you may notice that this chemical will appear negative. Okay, so it is safe, but this chemical will behave toxic. Okay, so if you train the model using the left chemical and you want to use the resulting model to predict the right chemical, it is likely that the prediction will be incorrect. So in the QSAR study, we call this activity cliff. Okay, so the possible solution is that you keep testing the chemicals with the same chemical scaffold. Then eventually, when you have enough data, you can find out what's the reason for these 2 chemicals to behave differently in animal toxicity models. But according to my statement in above slides, okay, so we don't have so much resource to perform so many testing. So we must find a solution for this kind of scenario. So if you take a look at these 4 chemicals against all these high throughput screening assays, they really behave differently, okay? So for this kind of low cost, high throughput external result, if you can make a good use of them, then we're supposed to improve the existing machine learning models for chemical toxicity. So this is what we try to do in the past 10 years. So this is a review paper that I published 4 years ago. So just to summarize what's going on in the drug discovery and chemical toxicity evaluation, right? So on the left side of this picture, for the simple chemical, physical chemical properties and some straightforward target binding, we have a good accepted models to make predictions for new chemicals. But on the right side, for animal toxicity, for human efficacy or side effects, so far we don't have very good models. Okay, but this is also the most important, the most interesting topic we need to start. So last year we published a review to just discuss the interpretable machine learning in chemical toxicity modeling. What people always criticize is you make a model, okay? So this model behave pretty well, make good predictions, but people don't really know how the model works. We give the name, call it a black box, right? So how the model, the prediction being made, it is also very important to evaluate a model. So there's two ways to do that. Traditionally, after the model being developed, we try to do search analysis to see which descriptors, which variables contribute the most to the final prediction. And then just go back to take a look of the chemical structures to see which part gives the most contribution to its toxicity. Okay, so this is after the model being developed. But in my lab, we performed this kind of analysis in another way. So before the model being developed, we try to teach a computer think like a human, right? To make the model in a smart way. So this effort needs the collaboration with toxicologists, needs the information from the public domain, for example, adverse outcome pathway information, and lots of other databases that we can find to make the computer think in a smart way. All right, so let's talk about the second part of my talk. That is chemical in vitro in vivo profiling. So we do have lots of data. So again, we are in the big data array. Okay, so how much data we have? So PubChem is the largest database for chemical information. So it hosts millions of chemicals testing against millions of assays. Okay, so if you go to the data counts page of this website, so this is February of this year. Okay, so you can see that about 100 million of chemicals being tested against about 1.6 million of bioassays and get almost 300 million data points. Okay, so if you download all this data to your computer, it is close to 10 terabytes. Okay, so this is so-called big data. It is not really easily handled by traditional machine learning or traditional data analysis tool. So you need a new modeling approach to deal with the big data. So in 2013, so this is there's a big project happened for that year. This is so-called Toxicology project, initiated by EPA. They use hundreds of different high-throughput screening assays to test hundreds of different chemicals. So before releasing the data, they released the chemical first. Okay, so for that year, I started my tenure track not very long. So my curiosity made me just use this, I believe, the total 900 chemical to search in the PubChem. Remember, this is 10 years ago. Okay, so there's no much data, like probably just like 5% of the current available data. But we still have lots of information for these 900 chemicals. Okay, so you see that on this direction, I list all the chemicals, but apparently it had been tested multiple times in different assays. And we have some data available, and of course, there's also lots of data gaps. And it seems that the people had different interests for different kind of chemicals. Okay, so for example, for pesticides, for drugs, people feel more interested. So there's more data available. And then for something like plasticizers, we have less data. Okay, so even before EPA used hundreds of assays to screen these hundreds of chemicals, actually we already have data available on the public domain. Just people do not like to see this public data. Okay, how to make good use of this public data. Okay, so we just did this project to do the modeling for acute toxicity. Okay, acute toxicity refers to the LD50. That's a lethal dose by 50% to kill the testing animal. So we have this in-house database of around 7,000 chemicals. We use 7,000 chemicals to search against PubChem, and you may see that the dimension of assays is similar to the dimension of chemicals. All right. So generally speaking, thousands of assays being used to test these thousands of chemicals. Well, of course, that most of this area agree, or means that that's also a huge data gap. But for this very massive initial data landscape, we already have like several millions of data points. Okay, how to make good use of this initial data? We use the concept of adverse outcome pathway. Okay, so what is so-called adverse outcome pathway? is when you have a chemical and this chemical is potentially toxic, it will bind somewhere you don't want it to bind. We call it offsite binding, okay? Then it will induce some kind of higher level responses, like for example, gene regulation, either it's upregulation or it's downregulation. Then it will make the cell response to this kind of regulation. For example, the oxidative stress. This is a very common toxicity responses of a cell, and it may cause the cell to die or have some kind of other mechanism like apoptosis. Cell toxicity refers to cell death. Then eventually it will cause some kind of in vivo toxicity. So here, what we refer to is the death of the testing animal. Then in the public domain, you may find the data for all these above steps. Okay, so for all these bioassays, you have the result, and based on the testing result of these assays, maybe you can find other chemicals with a similar chemical scaffold. All right, so it looks like that we could consider that all these chemicals behave similarly in a so-called toxicity pathway that will induce the final toxicity that we care about, that is acute toxicity, right? So here we can use this concept to select the assays that we think that is important, right? So for example, for the first assay, the initial binding, the chemical appeared active in this assay will majorly consist of these three chemical scaffolds. And for this assay, the later key event assay, it consists of the chemical appear active in this testing, majorly consists of these four chemical scaffolds. And for these assays, it consists of the active chemical with majorly these six chemical scaffolds. Then the last one consisting of these four chemical scaffolds. And you may notice that the same chemical scaffold appear multiple times in these assays, right? So we can use this as some kind of assay fingerprint to compute their relationships, okay? So we can construct this kind of relationship among assays. So if we calculate the relationship among assays, and we can use this as a similarity to describe their relationships based on the very messy initial landscape, we can create this picture. So here, each spot represents one assay. So if they color the same if they color with this same color, that means that this group of assays, we think that they behave similarly. They may belong to the same pathway. Okay, so totally how many pathway group we have is 45. Okay, so here if you let me show this. Anyway, so if the chemical, I don't know how to, yeah, here. If you see the chemical behavior in these assays, different chemical behave differently among all these assays, right? So it looks like that we have a pretty comprehensive profiles for the chemical that we feel interested and what we want to study. Okay. Obviously, not all these assay classes are useful to predict the activity, right? So we need to select those assay classes that are useful to predict the target toxicity that we feel interested. So what we use is the basic statistics, so-called 5-cross validation, to indicate that which assay classes has somewhat predictivity for the acute toxicity we feel interested. We find out that if we set up the threshold around like 60%, we have totally 25 assay classes have the certain predictivity for the target toxicity we feel interested. For those assay classes with this symbol, means that they are not statistically significant, means that they don't consist of enough data. Maybe the correlation just by chance, maybe the result is good, but we don't have sufficient data to prove it. Okay, so totally we have, in the end, we have 19 actually assay clusters that can be used to predict the acute toxicity that we feel interested. So let's take a look of some specific assay cluster. So for example, for this cluster 8, okay, it consists of totally 14 assays. And it has been used to test totally 63 chemicals. And among these 63, 56 true positives and 7 false positives. Okay, so it looks like that the predictivity is not bad, right? We can predict 56 out of 33. But since we don't have too many chemicals, only 63, then we can map these chemicals, 63 chemicals, among all the 7 chemicals in the chemical landscape. Okay, so just to take a look of these chemicals, and if you take a look of the chemical structures, interestingly, among most of the true positives, it consists of a very specific chemical scaffold like this. And none of the incorrectly predicted chemicals consist of this chemical scaffold. Okay, so if we include this chemical scaffold information into our modeling and into our predictions, then the predictivity can increase to 100%. Okay, and then my collaborators, so they are biologists. They like to organize the information into a pathway. So they use the assays that we identified to organize the pathway for oxidative stress. Okay, so oxidative stress refers to the important mechanism of this group of assays. Okay, so this is another class of assays. And among them, okay, so you can see that we can also identify a chemical scaffold majorly existing the true positive predictions. But among the false positive predictions, interestingly, we can say most of them consist of this kind of laddering chemical structure, right? So looks like that our chemical scaffold identification is not perfect. Okay, it does not include this kind of large ring structural information. So we can use this to improve our prediction as well. Okay, so since we do have so-called models, okay, although the models is not just one, we have like totally 19, right? So our collaborators, they give us a set of new chemicals, 623 new chemicals, not in the training side, for us to perform the testing. Okay, so if you enlarge the statistics, you can see that if we enforce the prediction confidence to 100%, so what does this mean? It means that all the chemicals must be tested against all the assays to get the prediction. Then the predictivity is 100%. But unfortunately, by this kind of condition, under this kind of condition, we only can predict 5 chemicals. Okay, so it means that the data gap is really large, right? So if we decrease the prediction confidence to around like 30% or 40%, then the predictivity of the resulting model is around like 75%. Okay, so this is also pretty acceptable. It's a nice prediction. So this is the paper published 5 years ago. So I think this is still interesting, so I want to introduce to you. But let's talk about what happened recently in my lab. Okay, so in the 2019, we also published a strategy paper just based on what we gathered in the previous 5 years. So at the beginning, we have a pretty messy initial data landscape. So we want to differentiate them based on the concept of adverse outcome pathway. As the molecular initial event and several key events, then the final outcome is the toxicity that we feel interested. Okay, so 2020, a year afterwards, we published another review paper just to summarize the 10 major characteristics of so-called big data. So these 10 characteristics can be summarized based on using the 10 words starting with V. Okay, so people are very familiar with the volume, with the velocity, but actually in the modeling study of using big data, you need to pay more attention to the veracity that refers to data quality and also the value of the data. Okay, so if the data obtained from very expensive protocol, then this kind of data, although it's still valuable, but however, it won't be very useful for modeling purpose. And this picture on the right, I obtained from Stanford University. The so-called big data, of course, it's very messy at the beginning. So you need to develop a method to sort it out, to arrange it into different categories, and eventually using machine learning to build a nice story that people can understand. Okay, so for the so-called machine learning, so the kind of popular method is the neural network, right? So what is the so-called neural network? At the beginning, we need to have a neuron refers to the output. So in our study is the chemical toxicity endpoint or chemical toxicity outcome of a model that we feel interested. And then we will have multiple input neuron refers to the training variables that we already have had for the chemicals in the training side. Then we use algorithm to find out the contribution of these input neurons to the final output neuron. Okay, so the weight refers to their contributions. Then in the end, we store this in a mathematical models and then use it to predict the new chemical. But currently, the popular way to do is the deep neural network. Okay, so between the input neuron and the output neuron, we have several hidden layers and it consists of 1,000 or even more hidden neurons. Okay, so the deep neural network is pretty popular and useful, but how it works normally we do not we are not very clear. Okay, because all these hidden neurons, so they behave like a black box. Okay, so we want to open this black box as well when we use deep neural network. Okay, so this is the concept of adverse outcome pathway and use the AOP concept to open this black box. Again, above matrix, okay, is the chemicals versus bioassays. Okay, so 1 represents that the chemical has an active result in this bioassay. 0 means that the chemical has an active result in this bioassay, and we also have some data gap. Okay, so appear at white. And for each column, this one single bioassay, we can make this as the just one neuron in this network. Okay. Then if a chemical appear active in this bioassay, it means that this neuron is activated. Same, okay, so if the chemical appear inactive in this bioassay, then we consider that this chemical is, this neuron is not activated. Okay, so for this neural network, each neuron has its biological meaning. It refers to one bioassay as a specific biological mechanism. Now we can also say that when we find out that this important path activated for the neuron first layer, then this one. So this path will be similar to an adverse outcome pathway that we want to identify. Okay, so we use this method to train a specific system data that we feel interested. So this is a estrogen receptor binding, which we specifically refers to the agonism, and the target toxicity, that is the eutrophic bioactivity. This is a one specific toxicity endpoint. Okay, so you can see that we can differentiate the assays based on their categories, initial ligand binding, protein stabilization, DNA binding, and et cetera, et cetera. And for the first layer, ligand binding, we have one assay related to the estrogen agonism. Okay, and for the second layer, we have totally 6 assays can be put it here related to this specific toxicity mechanism. And the third layer, we have 2 assays. And this one, we have 4 assays. And for the 5th layer, we have 1 assay. So we can train this model, and hopefully a toxic chemical will activate each layer only by we go through each layer only by the neuron refers to the assay that related to estrogen agonism. Okay, so here this is the training result. Okay, so you see that for the first chemical, it activated the assay in the first the neuron in the first layer refers to the assay that we wanted to go, and then three neurons that refers to the assay that we wanted to go. So all these paths are correct. Okay, so this highlighted relationship means that the contribution rank the most important. So we predict that this chemical is toxic, and we also show how it will induce this final toxicity by showing this pathway. Okay, so this is a pretty nice prediction with interpretable result. And for the second chemical, it's similar. Okay, so the only difference is for this layer, it activates another neuron related to estrogen receptor agonism, and not the same as the first chemical. Okay, so it shows pretty similar pathway, and it's also toxic. And for the third chemical, okay, so it's kind of different, because for the first layer, the only assay showing inactive result. Okay, so it cannot be activated. So in this case, it uses another assay showing active result to go through this network. But we know that this assay is not relevant to toxicity mechanism that we feel interested. But what does this mean? Mean that we do not have enough data for the first layer. Okay, so one assay is still not sufficient to cover all the offsite binding referred to this mechanism. Okay, so we need more assay for the first layer, right? So this is the third chemical. And for the last chemical, this is the only case in our training data, is you may say that it goes everywhere. Okay, so for most of the assays related to the estrogen receptor binding agonism, it shows inactive result. Okay, so for this specific chemical, it just goes everywhere to reach the final toxic outcome. And you can see that its structure is also very different comparing to the first three chemicals. Okay, so it behaves a very specific toxicity mechanism, cannot be covered by all these selected assays. Okay, so this is the only case in the training data. So since we do have a model, then again, we need to make a prediction. So we got a pretty large external database consisting of thousands of chemicals, and we can use our training model to make predictions. So here we must apply an applicability domain. Okay, so what is so-called applicability domain? It's very close to the prediction confidence that I showed in the previous project. Is I think we define that we need to have at least like 70 or 80% of the assay with the experience data. Then we think that the chemical is within the applicability domain. If we have less experience data in these assays, then it's out of the applicability domain. Okay, so you may say that if we have the chemical with sufficient experience data, then the prediction RC curve is pretty nice. Okay, but it's better than the chemical with less experience data. Okay, so this is the paper being published 3 years ago. All right, so then we aim on more complicated toxicity phenomenon, drug-induced liver injury. Okay, so for drug-induced liver injury, we choose this because liver is always the major target for most of the drug. And lots of drugs failed because of the liver injury. And some drugs even being withdrawn from market because of liver injury. So we have lots of data for that. Okay, and for FDA, it has a database called DILI rank. It consists of hundreds of chemicals. All of them the drug candidates reported to FDA. They have to include their liver toxicity results. So we have this pretty nice database as the starting data. But we also try to search literature to balance the toxic versus the non-toxic just for modeling purpose. Okay, so we use this database as a probe data to search against PubChem, try to find out which assay is the most important assay for dealing. So here, this is again, this is the initial data landscape. It's pretty massive, pretty similar to the acute toxicity project that I mentioned previously. Okay, but if we try to correlate these assays, all these assays to DILI, then we find out that this is the top 24 assays that has somewhat relationship with DILI. Okay, so this is the top 24 assays. And we select this ARE assay. So it's not ranked very high, it's here. So why we select it? So first, of course, this assay is important. I will show you why it's important. Second is just because our collaborator happened to have this assay in her lab. Okay, so she can perform experiment testing for us. So this is the reason we selected. So why ARE assay is important? This is the first high throughput screening assay being recognized by Tox21 program. Okay, and the paper published more than 10 years ago. So this is pretty famous. It can represent the ability for chemical to induce the oxidative stress. So the oxidative stress means that the chemical have the ability to induce the ROS, reactive oxygen species, in our body. So we know that all these kind of very active species are bad for our body. But how this ROS linked to the liver damage, we don't know. Okay. So, but however, the assay can tell you whether the chemical has the ability to induce the ROS in our body or not. Okay, so first we need to make up the data gap, because for the high throughput screening, although it consists of thousands of chemicals, it's still not large enough to cover all the chemicals in the DILI rank in the target database that we feel interested. So we can develop a QSAR model. So again, QSAR model may not be good for complicated toxicity phenomena, but for this kind of relatively straightforward mechanism, it still can be applicable. So in the end, then you may see that for the last model that we selected, the consensus here, the predictivity is around 75%. So here we use a different modeling strategy. We identify the structural alert considered to be related to DILI. Okay, so if a chemical, for example, consists of this very specific chemical scaffold, it appeared in totally 48 chemicals, then 73% of these chemicals showed as toxic to liver. Okay, so totally we have such as 27 such kind of alert. Okay, so we use a very straightforward definition for toxic chemical. So if a chemical has a structural alert, then it also shows the active result in ARE assay. Then we think that this chemical is toxic because the chemical is predicted to be toxicity by inducing the oxidative stress. This is how we define its toxicity. Then, if we only use the ARE assay to predict DILI, the prediction, of course, is not random. Okay, but however, the correlation is very weak. But if we include the structural alert into these predictions as the criteria I mentioned in the previous slides, then the result showed pretty nice statistic to predict liver toxicity. Same for the test side. Okay, so you may say that by using this strategy, it improved quite a lot. But still, we have a false negative and false positive. We have wrong predictions. So we need to say that what kind of chemical being predicted incorrectly. Okay, so here these four drug molecules, they actually are hepatotoxic, but in our model, it's being predicted as non-toxic. So they are false negatives. So we take a look of their responses, not in ARE, but in other toxicity assays. So it looks like that these four drug molecules, they induce activity in these assays. So it looks like that they have other toxicity mechanism, not just oxidative stress, to induce liver toxicity. Okay, of course, we still have false positive. Okay, so these two molecules, the top two drugs, they are safe for liver, but for our model, it's being predicted as toxic. So under this kind of scenario, we compare these two drugs with these three drugs being predicted correctly. Okay, so because they are drugs, we can have is Cmax, the maximum total concentration of the drug, means that the availability of this drug in our blood. So the concentration of these first two drugs in our blood is pretty low. Okay, and it's not reach the concentration that needed to activate to be active in ARE assay. Okay, so if you see that it's much lower than the concentration needed in the ARE assay. And for the later three drugs, their concentration in our blood is high. It's sufficient to reach the concentration needed for ARE activity. Okay, so looks like this is the exposure problem, right? So when we perform the modeling, exposure needs to be considered to make a better prediction. So since we have a model, then we can make predictions. Then our collaborator can test the chemicals in the ARE assays. So first we select 14 chemicals in the test site. They have the daily result, but however, they don't have the ARE testing result. So we use our QSAR model to make predictions. Then our collaborator tested it, it's 100% accurate. And when we correlate it based on our strategy, it predicted daily also 100% correct. Okay, so we are really happy. Looks like we solved the problem, and the model behave 100% accuracy. But unfortunately, this is not the case because these 14 chemicals came from the test site. So then we select new 12 chemicals from literatures, from drug bank, from other public sources. They have the daily result, and they are not included in our training data and the test data. This is the actual condition to predict the new chemicals. Our model can predict around like 70–75% accuracy. So we still have some wrong, incorrect predictions. So we have lots of inconclusives, cannot be classified. So this is the actual condition. Okay, so this is the paper published like 2 years ago. What happened this year is we use the combination of ARE and another assay, the mitochondrial disruption assays, then to see whether we can improve this model prediction. Yeah, we can make another assay represent a new mechanism that the model performance can improve, but still cannot reach to 100%. Of course, that the delay is much more complicated. It's not just the two mechanisms. All right, again, according to my statement, exposure is very important. Okay, so all the drugs are toxic if the exposure is high enough, right? So the dose responses information need to be considered, and it needs to be integrated into the modeling. But the difficulty at the beginning to use this kind of data is we have a different dose response curve for the same chemical for the same assay coming from different resources. So for example, here we show that this chemical has 3 similar but different dose response curve. Actually, here is concentration response curve from 3 resources. Okay, so this is pretty common for the public data. But to use this kind of public data, we need to find a strategy, right? So we created this kind of so-called pseudo dose response curve. It merged the information from these 3 curves to form a new curve. Okay. So here, I just want to mention that for these 15K event assays, we merge 59 assays, we merge 18 assays, and here we merge 6 assays. They are the same assay, but however, they just have a different dose response curve, a concentration response curve. So we need to treat them first. Okay, so we have a new dose response, that's concentration response curve. First use to this is to run chemicals, right? So for example, using this assay, ER antagonism assay, the concentration response curve or dose response curve, we can rank chemical like this. For the highest ranked chemical, these chemicals show not just high activity, but also high potency. And for this chemical, on the contrary, okay, it shows the low activity and also low potency. And similar for this assay, if you rank the chemical, then this chemical is highly active and also highly potent. But for this chemical, obviously, is inactive and not potent. Okay. Oh, and of course, that for these two chemicals, they are likely to be toxic, and these two chemicals, they are likely to be non-toxic. So this is the first use of this information. And the second use of this information is we can see the cluster of chemicals. So for example, if the chemical consists of this common scaffold, then they always appear active or ranked highly in all these assays. Okay, and the chemical with this chemical scaffold, they also have a very specific toxicity mechanism. And again, so we want to use this to incorporate with the exploratory information. So this is the protocol developed by EPA called HTTK. They use Monte Carlo sampling and also high-throughput screening assays to generate the so-called steady state concentration. That is the concentration in adult for specific chemicals. So, for example, for this chemical, first chemical and second chemical, if you compare these two chemicals, they are pretty active, right? But the first chemical exposed in human also highly. So this highlighted region cover all the dose response curve or concentration response curve needed for generated from the assay. So obviously that this chemical is likely to be toxic. But for this chemical, on the contrary, it is being exposed in human in a very low concentration. Although this chemical is pretty toxic, but however, this concentration does not reach the concentration needed for its activity in all these assays. So this chemical, on the contrary, is non-toxic. Excuse me. So we integrate this information together with all these key event assay ranking, and we can differentiate the toxic chemical versus non-toxic chemical from hepatotoxicity data set pretty well. This is a paper published last year. Okay, just to summarize what I talk about today. First, the data is of course very important. So we have a big data definitely at this moment. Just the data pretty messy, pretty big. So we need a new approach to be developed to handle this kind of very messy, very big data, and make the model interpretable. Not just make a prediction, but also make the prediction interpretable. And through this procedure, we need to collaborate closely with toxicologists, and then make the computer smarter to train all these models. Then in the end, I also want to introduce the Integrated Transporter Elucidation Center. This is a new founded research center among 5 universities, okay? Rutgers, Rochester, University of North Carolina, Tulane, and UPenn. So under this center, we have totally 3 projects, okay? The first project is try to define different kind of transporter proteins in placenta. And I'm leading the second project is to develop machine learning models for chemical bindings, especially drug binding to these transporters, and use the profiles of the transporter proteins in placenta. This information generated from the first project to predict the drug safety in pregnant women. And then the third project is to use the ex vivo data. So the protocol they developed, so-called placenta on a chip in UPenn group, to evaluate the proposed drug molecules by us. So our totally, we have 3 projects, and also the project work pretty closely to each other. So if I have a chance next year or in the future, I will also be glad to present what being generated through this pretty collaborative and big research events. So in the end, I also want to thank my group. So all this research project being finished by my student. What I present today majorly is being done by 4 of my peer students, Danny Russo, Heather Cialele, Xuelan Jia, and Elena Chen. And the senior collaborator in this project is Judy Strickland from the CDM, and also the experimental testing were being done by Laura Alexanis and Winsha from Rutgers, New Brunswick. And of course, in the end, NIH and NSF gave me funding to do my research. Thank you very much, and I welcome any kind of questions.
Dr. Zhu Hao, thank you so much for your inspirational talk. I think I learned a lot from you. You know, I will watching people who are raising questions. Before I see that, I will ask you quickly. So my first question is really about the toxicity outcome data. When I look at you present your work, I think sometimes I saw animal model-based toxicity. Sometimes I feel is based on human data. Can you go there a little bit more?
Oh yeah, so generally speaking, I didn't really specifically mention which data is from human, which data is from animals. Yeah, so so for the RD50, so the diesel dose, of course, we cannot test it on humans. So this is animal data for sure. So for this 7,000 chemical database, but for the daily rank, it is human data. This is from clinical research, and for the eutrophic activity, this data this is from animals. It's not from human. Yeah. So for the hepatotoxicity predictions, so I didn't really mention for the so the daily experiment. So here, when they come from the daily rank database, yes. So this is from most of all this data from human, but we also use some animal hepatotoxicity result to test our model. So I actually forgot the details, but we also in the among this project, we also use some animal data for the test purpose. Yeah.
Got it. I put it this way. So I think one of the purposes of having your talk is really looking for collaborations. I think that's where we can figure out a way to collaborate.
Yeah.
I think we are focusing on clinical data. I think not only adult population, but also pediatric maternal population. We look at literature, really is clinical data. Then in the meantime, there's a lot of animal study published as well, right?
Yes.
So we also look into that as well. I feel it's going to be critical to figure it out. What will be the animal toxicity data, what will be the clinical toxicity data, and how consistent they are. Because I think from phase 1, from preclinical to phase 1, there's a huge gap over there. How do we do the translation, right?
Yeah.
I feel that is there's huge need in terms of figure that out.
Yeah.
I'll definitely follow up with you on that. All right.
Yeah.
Okay.
So
Fantastic.
I feel very interested because first we do not have too many clinical data for sure. This is the kind of short disadvantage of my research is we just work with the public database, and for public database there's not many clinical data. Yeah. So we definitely want to use more clinical data in our model.
Absolutely. And let's see who else is raising a hand. I haven't seen much. Let me see anybody raise the question. Not much in the question and answer session as well. So let's do this, Dr. Zhu Hao. Let's talk offline, and I'll reach out for probably plan another meeting, Zoom meeting
Mhm.
just between you and me, and trying to figure that out. Okay.
Sure, of course. Yeah, I'll be glad to. Yeah. Yeah,
Uh,
my talk is actually about chemistry, so I think that's the reason most people do not
no, we will
want to
love
ask
it.
questions.
We will
Yeah,
love it.
yeah,
We will love it.
yeah.
And thank you so much for spending time with us. Looking forward to talk to you more. Okay.
Yeah. Let's follow up for
Mhm.
possible collaboration. I'll be glad to. Yeah.
Okay. Good. Good. Thank you so much. I will keep in touch.
Sure.
Bye bye.
Bye bye.